Well, welcome everyone to the Monsoon Twilight briefing. Thank you to FB Rice for hosting us here once again. So my name's Mark Parsh, I'm from BioShares. I'm editor of the BioShares newsletter. It's been going on for 23 years, and I noticed recently that a drug we wrote about in edition 1 recently got approved, the one from Chris Burns and Andrew Wilkes. So that's how long I've been doing this for. So we're being recorded here today. We're going to have 3 companies present, quite different companies, so better have your thinking hats on. They're going to present for about 15 minutes, then we'll have some Q&A, so have some questions for them. Now I had a couple of them are Perth-based companies, or were, but they're now no longer based in Perth. The CEOs are in New South Wales and Melbourne, but I had some Perth jokes, which I was going to say, well, they've come over here to collect their #1 draft pick, Harley Reid, from last night, but and well, they've come here for some good coffee. I don't know, but can't say. I just got to throw all those jokes out the window. So welcome. So difficult time for biotech still. I've been saying the same thing for a while. Everyone knows it's been pretty tough out there, but I think we are starting to see some signs of a bit of movement in the sector. If we look at some companies like Lumos Diagnostics, which shooted up about 1,500% on some FDA approval. A couple of weeks ago, Imugene was up, I think, by about 200% or so almost on some phase 1 data. So I think it's been a bit of a bruised and battered sector. But this sector does tend to go in what I believe are 5-year cycles, having sort of followed for almost 24 years. And Jeremy from IB Bioscience Managers, sort of I was telling him in the lift that he agrees that it does tend to follow these sort of, broadly speaking, 5-year cycles. And if that's the case, then the last cycle started 2020, then maybe we're a year away from the next uptick. So perhaps sometime next year we'll see some an uptick in the sector, and with interest rates plateauing in the US and possibly declining being the next move, that may prompt some interest in this space. So get set for your biotech companies, get to know them well. So the first company is Arovella, which is involved in CAR-T therapy. Now we're going to have Michael Baker talk to us about Arovella. Michael joined Arovella in 2020. It was previously based in Perth. Michael's now based in Melbourne. Second person today that I'm talking to who was an investment manager or analyst and is now working in the biotech sector. So please welcome Michael.
Do you have the little clicker?
Yeah, it's just on the right.
Oh, great, thanks. Great, thanks, Mark. Thanks everybody for joining us today. It's great to have you here. It's a lovely day outside, so I appreciate you taking the time to be indoors listening to us. And I'll go quite quickly because we do have only 15 minutes, and I would like to take the time to answer some questions. So I will be making forward-looking statements, so please do note the disclaimer. And I'll start with the highlights of the company. We are working on an invariant natural killer T-cell platform, and I'll explain what that means in a moment. But in short, it gives us opportunities for targeting therapeutic or cancers like blood cancers as well as solid tumours. Our lead product is called ALLO-101, which we're hoping to take into the clinic next year. We do believe that our iNKT cell platform addresses unmet needs that currently plague the cell therapy sector. We have assembled an exceptional team, both at board level and management level, with a lot of experience in drug development, particularly cell therapies. And the way we've built the company is through a series of strategic acquisitions for IP, which we've taken from some of the best research institutes and universities in the world. So we do see that for all those reasons we are in a unique position as one of a handful of iNKT cell companies globally and the only in Australia. So let me quickly touch on cell therapy and what it is and why we're excited by it. So CAR-T is what it's probably more affectionately referred to, and I'll explain why we're different from that in a moment. But in short, this has enabled us to use the words cure and cancer in the same sentence, which is a lovely place to be. And the reason that we're able to do that is that patients have taken these therapies experimentally and beyond and are living more than 10 years cancer-free. So not surprisingly, the revenues have picked up already for the first approved products. And of course, we're seeing that this is going to be an enormous market, predicted to be more than 60 billion by 2030. Notwithstanding, 40 to 60% of patients that do take these therapies do relapse, so it's not curative for everybody. just a very quick example. This is a young lady, Emily Whitehead, that took the first CAR T dose experimentally as a pediatric patient more than 10 years ago, 11 this year actually, so she's now still 11 years cancer free, which is remarkable. So how does it actually work? And just I want to make it clear here, this is really the ultimate personalised medicine. So imagine on the very far right where we're dealing with Emily Whitehead's blood. So it actually comes from the patient. You can see in the top right, we take Emily's T cells. They get genetically reprogrammed in the laboratory somewhere, and I will point to this side. On the surface of the cells, we have what's called a chimeric antigen receptor, or CAR, and think about that like as a beacon or a homing or a guided missile that will actually direct the immune cells towards the cancer and trigger their destruction. They get grown up into the millions before being injected to that same patient from where they actually started from. So this is important. It starts from the patient and finishes with the patient. So we believe that actually poses a significant challenge for the sector. So on that top right scheme, top left on your side, so we see that it's one therapeutic for one patient, okay, and that's really ineffective from a supply chain and logistics point of view. And what it does is it means the dose costs about $500,000 per person US. So we believe that that's problematic, obviously, and I'm sure payors will believe the same. In addition, because we're taking it from the person that's seeing the cancer or chemotherapy, their immune cells generally aren't doing that well, so we have a compromised starting material. And also, not all treatment centres can actually manufacture these therapies. So there's a lot of patients that will actually be eligible to take the therapy and they don't actually receive it. So what we're working on is our iNKT cell platform, and this is the hope that we have a single donor, a very healthy 25 to 35-year-old person. We take the starting material from them and we're able to make the drug so we can actually give it to multiple people. So another disadvantage of CAR T is because it's made just in time. It takes about 4 to 6 weeks to make it on average. People are working hard to bring that cycle time down. Irrespective, what happens is patients can actually die while that therapy is being manufactured. So again, not an ideal situation, versus if we're able to pull it out of the freezer, we believe that's a great place to be because we can give it to a person in hopefully less than a week so they're able to take it if they're eligible. So what is it exactly that we're looking at? And as I said, it's iNKT cells, and it's invariant natural killer T cells is the full name. So this is just a handful of immune cells in that diagram that people have studied in relation to our immune system. So T cells on the top right, NK cells in the bottom in purple. They've been the most well studied, and what we're interested in the middle there is the iNKT cell. So they're naturally occurring immune cell in our body, but why we're interested in them is they don't cause what we call graft versus host disease, which means they don't attack a person once they've been given from one person to another. And this is what enables us to create the therapy from one person and give it to multiple people. In addition, they are the front line of the immune system, so they naturally work to remove infectious diseases and also certain cancer cells. They cross both sides of our immune system, the fast-acting and the more durable immune response, and they actually naturally target cancer cells that have something on their surface called CD1D. So in addition to that, they actually have a range of other anti-cancer properties as well that we think are quite important for solid tumours. So I'll skip over this because it's a diagrammatic form of what I've just said. And just looking at the manufacturing advantages here. So as I said, we start with a healthy individual. We collect their iNKT cells, we take them to the lab and genetically reprogram them, and then we expand those up into the billions. So now we've got lots of these iNKT cells with the CAR or the guided missile. They get stored, frozen, ready for use, and that when a patient's ready to take it, they'll be thought out and utilised. So again, big advantages we see are it's healthier starting material because they haven't seen cancer or chemotherapy. It is scalable, so we have multiple batches or multiple doses per batch. It gives patients faster access to the treatment, and because they're stored in a freezer, there is no opportunity for manufacturing run failure. So very quickly on the commercial side of things, looking on both sides of the slide here, we've got T cells and NK cells. So these are a snapshot of the different players in each of those spaces. There are hundreds of companies and thousands of clinical trials ongoing. And looking in the circle in the middle, this represents the total iNKT cell landscape worldwide today. So we're one of a handful of players working on this particular immune cell type. So we do see that we've carved out a really important niche in a very exciting area of cancer research. So let me just talk about our lead product, ELA-101, briefly. So it's what we've termed a next generation and off-the-shelf therapy for what we call CD19 positive blood cancers. So I won't go through this in detail, but suffice to say the blood cancer market is quite large. There are hundreds of thousands of cases per year, multiple deaths. CAR T products have had success and they are moving up to second-line therapy, but there's still no off-the-shelf CAR or cell therapy that targets blood cancers that has been approved worldwide. So just talking through some of the data here, this is some experiments that were performed and published in a prestigious journal, Cancer Cell. And what we're looking at here are a series of mice on the top 2 rows. It's day 0, so they've actually been engrafted with a particular lymphoma model. So anywhere you can see the colour in those animals, that's actually the cancer cells inside of the animals. And then down the bottom, we're actually looking 3 days after treatment. So keep that in mind, it's 3 days, so it's quite a short time period. And in the first column, it's essentially salt water. The second is unengineered T cells. The third is unengineered iNKT cells. And then the fourth is the equivalent, or closest equivalent to what's approved and in market today, a CAR T. And then the last column is Arovella's Ella101. So what you can see quite clearly, in the bottom 2 rows, you see there's quite a lot of cancer cells persisting in the control groups, but quite clearly there's been a strong eradication of cancer cells in the iNKT cell treated animals. So great that it works quickly, but we need to know that it works over the long term. That's what we're most interested in. So this is a very similar study design, only now we've got a lot more animals included in this particular experiment. On the vertical axis, we're looking at how many animals are actually alive. So 100% would be all the animals alive, down to 0%, which is all the animals have died. And the horizontal axis, we're looking at 90 days for this experiment, so a much longer time frame. So all the controls, black, blue and green, they have all died in the order of 40 to 60 days. The orange line is the CAR T animals, so you can see there's about 60% of the animals are alive at 90 days, which is really good data, and that would have led to approvals for some of these products eventually to go into clinic and obviously to get approved and in market. But when we look at the red line, it's quite clear that the iNKT cells do perform better than the CAR T equivalent. Now this is just one more finding that we were quite excited by here. It's 4 of the 19 mice that were treated with our therapy. They actually look, you can see this is the 4, 1, 2, 3, 4, and it's a time series for each. At the earliest time point, they looked like all the cancer cells had been eliminated. But about the 20 to 30 day mark, you can see the colour starting to reappear, and it's coming back really specifically in the brain, which was confirmed. And so the natural thought here is they'll die, and that will be the end of, or the data on the previous slide won't look as nice. And what happened instead was the animals actually underwent what we've termed a spontaneous secondary remission. So the cancer cells were actually removed a second time with no further dosing. So it suggests that the iNKT cells are able to remain and eradicate the cancer cells a second time. So very quickly on this program, so where we are, we've demonstrated already this approach has robust potent activity or anti-tumour activity. What we're expecting is that the therapy will be safe. iNKT cells have been used in clinical trials. The CAR or guided missile targeting CD19 has also been used and it's an approved product, so we're not expecting safety issues. And we're working through the manufacturing where we're able to make multiple doses per batch. So the next step is aiming next year to take this therapeutic into clinic. So I'd just also like to pivot a little bit and talk about our approach to solid tumours and why this is important. Approximately 90% of cancer diagnoses are some form of solid tumour, so we do think this is a pretty important area to target, both from a societal perspective but also a commercial opportunity. And the 3 strategies we've got on the far your left, it's to license novel targets, which we've been doing. The second in the middle is to armour the iNKT cells, so we actually improve the platform overall. And the third is to work with unique groups and create partnerships, so we're actually combining therapeutic strategies. So for the first strategy, we announced about 6 weeks ago or so that we actually in-licensed a novel guided missile or CAR from another company based in the US. It's termed Claudin 18.2. The really nice thing about getting these new targets that direct the cells to different cancer types is they can actually now fit into our workflow for manufacturing. All that we need to do is, imagine this is the CAR that we would like to get in. All we need to do is get a new lentivirus, which is something we can get a manufacturer to produce, and that goes into the workflow. So we've worked very hard to create this manufacturing pathway, and we'll continue to leverage that for each product that we continue to develop. So why are we interested in Claudin 18.2? It's become a very exciting target across the oncology sector. Some people saying it's as important as HER2. We know that it's also expressed in a range of different cancer types like gastric cancers and pancreatic cancer, and it is a validated target. So there is a therapeutic, a monoclonal antibody, which is a type of therapeutic strategy that will be approved in January 2024. It extends lifespan by 2.5 months, and it's predicted to have peak sales of 600 million to 1.3 billion. So we're quite excited to have a cell therapy, which we would expect to have better activity than a monoclonal antibody going after this target. So not surprisingly, the gastric cancer market's also set to be quite a large market by more than 10 million by 2030s, but that's just gastric, not accounting for all the other cancer types where this could be applied. So the second strategy, as I mentioned, is to armour the cells or enhance the cell platform itself. So we're doing that through use of what we call a cytokine technology, and we have an option for this particular technology already from the University of North Carolina based in the US, and that actually was optioned from one of the inventors of iNKT cell biology, so very well credentialed. And then again, similar to the targeting strategy, we can incorporate this technology into our manufacturing workflow so we don't have to repeat a lot of what we've already done. So in terms of how this works, we presented some data earlier in the year. We've been able to show that it does actually work by increasing the number of iNKT cells that are present in the animals to the tune of 10 times more cells actually present in the animals and actually doing what we want them to do. And as a result, the hypothesis is that leads to better anti-tumour activity. And that's exactly what we've seen for a particular solid tumour. In this case, 75% of mice treated with our iNKT cells that contain that technology are alive at 61 days versus 0% without that technology are alive at 49 days. So we know it has the benefits that we want, and if we are happy with our due diligence, we would look to in-license that into the company, integrate it into our platform. So the last strategy, as I mentioned, is to work with innovative biotech companies in Australia. So we're working with a group called Immugene, who have a very neat oncolytic virus. And what they do is that virus has been developed to essentially infect solid tumour cells. And once it's done that, it will naturally divide, replicate, and burst some of those cells. But for the ones that it doesn't, it actually tags them with a little green bump indicated here that's called CD19. And our iNKT cells will recognise that CD19, so they'd be administered second. They come in over the top, and they can destroy any of the cancer cells that haven't already been eliminated. So excited by that program. We had good in vitro data earlier in the year, and looking forward to getting in vivo data before the end of this calendar year. So we do have an expanding pipeline. As I said, we've got our ALA-101, which we're developing for blood cancers, non-Hodgkin's lymphoma, and so forth. We're partnering with Immugene. That gives us a range of opportunities to target a variety of solid tumours. And as I said, we're looking to bring in novel targets like Claudin 18.2, which also gives us a range of shots on goal for solid tumours, and also integrating our cytokine technology to enhance the platform overall. So very quickly, the milestones for fiscal year 2024. Big one for us is to take the product next year into clinic. So what we're working through at the moment is optimising and scaling up the manufacturing before first half next year, completing a manufacturing run to then be in a position to create all the regulatory documentation for submissions to the FDA or TGA. And for the preclinical side, getting data for the Immugene study, as well as the looking to in-license the cytokine technology if our due diligence is appropriate, and of course generating proof of concept studies, data I should say, for the additional preclinical programs that we've got underway. So very quickly, I'll just touch on our financials. So currently capped at about 80 million. We have, at the end of September, we had 5.3 million in the bank. We just got our R&D tax rebate a couple of days ago, which was 1.9 million or so. We're very happy with our register. We've got a range of supportive investors on the register, like Merchant Group, as well as another range of high net worths and smaller institutional investors. But we're, unlike a lot of the biotech sector this year, we're delighted that we've managed to buck the trend, which has been pleasing, and we're looking forward to endeavour to continue that over 2024. And I'll finish here very quickly. So this is just, again, I think this helps, obviously for someone that's developing a cell therapy. You can see there's quite a range of transactions that have continued to take place in the cell therapy sector, some only a few weeks ago. and they are sizable upfront payments. There's some in the order of hundreds to $200 million payments or equity investments upfront. And again, I think that just shows us that we're in a very exciting sector. There's still a lot of investment. You can see there's quite a number of repeat offenders on that list, like AstraZeneca, Genentech, and so forth. So we're still expecting this to be a really important part of the ecosystem moving forward. So very quickly to summarise, we have an exciting, novel, off-the-shelf iNKT cell platform. We think that will improve a lot of the manufacturing and logistics challenges that plague the cell therapy sector. iNKT cells, CAR iNKT cells I should say, have multiple properties that we think are really beneficial for both blood cancers and solid tumours. We're excited that our lead product is advancing through the manufacturing stages and looking forward to take that to clinic next year. We continue to expand the platform and we'll continue to make that a priority, not building sideways, but building towards a very strong targeted platform. And we think for all these reasons that we still sit well positioned for growth. And with that, I'd just like to say thank you again very much for taking the time and for your attention. I'd be delighted to take questions.
Thanks, Michael. Do we have any questions for Michael? One of you.
Thanks for that. Not to digest. I realise it's early days, but is there any reason why the Cloud 18.2 could not be used for the OncoLeaks program imaging instead of the CD19? Is that something that potentially is a synergy?
Yeah, absolutely. Look, that's a good question, and there's two ways you could go about it. Do we actually think about putting one of the other targets into the oncolytic virus that iNKT cells always recognise? So no matter which CAR we use, we could combine it with Immugene's oncolytics platform, or just say switch out the CD19 for Claudin 18.2. So I kind of like the idea of having even us having a dual CAR, so we could have a CD19 and a Claudin 18.2. And if you think about it like a Venn diagram, a lot of the tumour cells will have Claudin 18.2. The ones that don't, force them to have CD19, and then you've just got hopefully a good representation of the entire tumour.
Mark, how are you going?
Good. Immunology. Do you know anything about immunology, Mark?
Just a little bit. Mark Nash, Melbourne University.
Nice
so,
to meet you.
interested. I'm not totally familiar with the work, but what mouse model are you using there in those earlier photos?
Yeah, so it's an NSG mouse, so they lack the functional immune system.
Right.
But we have a lymphoma model that produces both CD19, and I didn't talk about it for brevity, but also CD1D, which is a natural target of iNKT cells. So while we wanted to do that as well, is that both of those targets are expressed on a few human lymphoma models.
Second question, what cytokines are you loading your vector system with?
We're not saying that just yet, but stay tuned.
Very secret.
Yeah,
And Michael,
it's just patent issues and so forth.
which company of those competitors? Which company is probably the leader in the iNKT space?
Look, it's probably Ming, just by virtue of the fact they've got a product in clinic. Having said that, I think they've gone with a product that will may have a difficult time. It's unengineered iNKT cells, so it doesn't have the guided missile to strengthen it against cancer cells. So they're doing that in solid tumours, and there's a few other things that they're not doing, which bypasses the requirement for any chemotherapeutic intervention up front, which is it's a good goal. I think it's, as I said, it's setting the bar for them really tough in terms of what they might see. If they see good data, that's phenomenal. But what they will get out of that is excellent understanding and information about so I didn't say that, but in that trial they're giving 1 billion iNKT cells, so 1 billion, it's a huge amount, from a healthy donor to 32 unmatched patients, and so we're able to see no GVHD. So that's great. That's a really important piece of data that we can leverage as well.
Okay, please take Michael.
Sorry,
One more question.
one more.
Yep.
Are you planning to do your phase 1 in Australia?
We haven't set it concrete where we're going to do it, but Australia is obviously a very attractive place for the R&D tax rebate. So we're manufacturing our product at Cell Therapies, which is embedded in the Peter Mac. So I think there's a lot of strengths that we could leverage, but certainly our focus will be to look external and to markets like the US through the FDA.
Oh, thanks.
Thanks.
Right, our next company presenting is Pharmost. and CEO is Michael Thurn. Now, Michael's a very experienced drug developer. I'm not sure which companies you were from, but I know you've taken a few drugs through. I know were you at Cytopia?
Cytopia.
And
Spinifex.
Spinifex. And you got those drugs through, or the Spinifex one through to the help get through to the market?
No, not through the market.
Not sorry, to registration.
No, it's not through registration.
Oh, is that still going?
Yeah.
Yeah. Okay.
So Spinifex was acquired for 700 million
Yeah.
at phase 2
Yeah.
by Novartis.
Okay.
So it hasn't quite got there yet.
And so Michael's now CEO of Pharmos, which is doing a bit of everything. Human and animal cancers, and also treating motor neuron disease. I think the motor neuron disease, is that more of a recent sort of application of the technology?
Yeah, so about 2 years. Yeah.
About 2 years, okay. All right, so please make Michael welcome. Thanks.